See that 8 cell stage is here. Furthermore, blastula is here, which is a ball of mass, and if you make the cross section of this blastula, you can see that there is a cavity present inside this cell of mass, which is called as blastocele. In some organism, the body cavity develops from the blastocele only. That is not considered a true body cavity or true coelom cavity. So this is the blastocele, and if you see that in the further process of development, in the next stage, which is called as gastrulation, the layer of the cells invaginates towards the blastocele cavity, and in this case a opening is formed which is known as blastopore. So how does this blastopore opening affects? How does we categorize organism into the two category based upon this blastopore? We will see in the upcoming slides. So on the basis of the germ layer formation, that means whenever a zygote develops, so usually in the previous slide you can see that the ball of mass is there. This cell of mass, so this mass of cells is basically the cells which are undifferentiated cells. So these cells are not differentiated for a specific kind of function or for a specific kind of role. So what happens during gastrulation period, these cells usually differentiates. When they starts to differentiate, they form the germ layer. So there are three germ layers: ectoderm, endoderm, mesoderm, right? So on the basis of the presence of three germ layers, many organisms are categorized into either diploblastic and the rest are categorized into the triploblastic. So diploblastic are those organisms which are having two germ layers, that is ectoderm and endoderm. So the invertebrates, lower organisms are usually diploblastic. They are only having two germ layer, ectoderm as well as endoderm. Mesoderm is absent in those organisms, particularly in diploblastic. And in triploblastic, all the three germ layers are present: ectoderm, mesoderm, endoderm. And we have to keep in mind that whatever the organism's body is forming different tissues and different organs, these all are formed from these germ layers only, whether it is in diploblastic or whether it is in triploblastic, right? Some examples are also there mentioned, like coelentrates, ctenophora again. So these are diploblastic. Porifera is cellular aggregation, so there will be no tissue differentiation, no tissue formation, there will be no germ layer, so it will not be categorized into the either diploblastic or triploblastic. So here we can see in the triploblastic, you can see that from platyhelminthes to chordates, all are triploblastic without any single exception. So as we move further, you can see that if we arrange these layers according to from periphery to the inner side, we can see that outermost layer is formed by ectoderm, the innermost or you can say the gut cavity is formed by endoderm, and between the ectoderm and endoderm comes the mesoderm. So during embryonic development, ectoderm and endoderm forms first, and then after mesoderm forms later on in the triploblastic. In the diploblastic, mesoderm will not form, so they will remain without mesoderm. So here we can see that in diploblastic particular, this outer layer is the ectoderm, right? The inner layer is the endoderm. This is the gut cavity. Here also it will be the gut cavity, same. And in diploblastic, as mesoderm is not present, so the space between the ectoderm and endoderm is filled by a gelatinous layer known as mesoglia. We must remember that mesoglia is a non-cellular layer. Like we can see here, the ectoderm and endoderm both are cellular layer. So mesoglia is a jelly-like layer in which cells although are present, but in fact it is a non-cellular layer, right? So in the diploblastic, mesoglia will be present, or in the case of triploblastic, we can see that mesoglia is not present. Instead, mesoderm germ layer is present, it is formed. So all the three germ layers are present, hence it is called as triploblastic. Now, as we move further, and as I mentioned in the previous slides that when the gastrulation process occurs, the blastopore opening forms. Depending upon the differentiation of the blastopore, that in the future or in the embryonic development further, this blastopore, if it forms mouth opening first, then it is called as protostomiates, or if it is forming anus first, then it is called as deuterostomiates. Here we can see the diagram here, that if the blastopore is forming mouth first, anus later, then it will be called as protostomiates, and the organism are categorized into protostomia. Or if it is the vice versa case, that means blastopore is forming anus first and the opposite end is forming mouth later, then it will be called as deuterostomate. So here we can see that when this develops, this cavity is called as archentron cavity. I can write it down, archentron cavity, right? And if the blastopore is forming pore, like here we can see that anus develops from the blastopore, this side, and here this is again shown as anus, but instead it is mouth in the case of deuterostomiates, and in the vice versa case, protostomiates, it will be mouth and the opposite end will be the anus, right? So in this way, the protostomiates and deuterostomiates are forming. Here we can also focus on the coelom development, as we can see, as we will also see in the upcoming slide also, that mesoderm is present as a mass of cells here during gastrulation process. This layer is the ectoderm, this layer is endoderm, and this is the mesoderm. Okay, so I will also share this same picture in the coelom context. So I'm giving you a little brief introduction that this mesoderm mass, if it develops into the coelom, then such type of coelom is called as schizocoelom. And if the gut pouches is forming mesodermal mass, mesodermal pouch cavity, then development of such kind of coelom will be called as enterocoelom. So we will discuss this also. So coming back to the next point, coelom. So starting we have discussed the blastopore development, then the germ layer organization, and now coming back to the coelom. Coelom, students, can be defined as, I have already discussed this in the yesterday's slide, that coelom is the body cavity which is enclosed by the mesoderm germ layer. So the primary requirement is the presence of a body cavity. The secondary requirement is it should be enclosed by mesoderm germ layer. Then and then only we can term it as coelom cavity. So this is nothing but the NCERT diagram which displays the three types of category of coelom. That means first one, which is not having any body cavity, it is called as acoelomate. The second one, which is having cavity but present in the form of pouches scattered in the organism's body, that will be called as pseudocoelomate, or organism will be called as pseudocoelomate and the cavity will be called as pseudocoelom. And then comes the true coelom, in which the cavity is enclosed by mesoderm germ layer. So here we can see the mesoderm is present at both the sides and the cavity is enclosed by the mesoderm derived germ layer. So that is why we are calling it as true coelom, right? So as we have already discussed, the triploblastic animals could be further categorized into three types: acoelomate, pseudocoelomate and eucoelomate. Eu means true, coelom means coelom. So true coelomates can, on the basis of the further development, as I have mentioned in the previous slide, schizocoelom and enterocoelom, they can be categorized into schizocoelomate or enterocoelomate. Okay, so let us discuss this further. First of all, starting with the acoelomate. Now in this chapter, the diploblastic are acoelomates, definitely, because they are not having mesoderm germ layer. If you look at the NCERT, Porifera are also categorized into acoelomate because it is acellular, because it is having cellular aggregation, right? So starting with the first category, triploblastic acoelomate. Now, in this, the body cavity is absent. Example, we have written platyhelminthes. Now, during embryonic development, if the body cavity is formed, it is filled by some space filling tissue, which we can call it as parenchymal tissue, or in some books or module you can find it as a mesenchymal tissue, right? So parenchymal tissue is a tissue which fills the space or which fills the cavity. So in that way, there is no cavity is present. So here you are not seeing the space. So if you see the actual diagram, it is like this, that the cells have completely filled out the cavity, and therefore as a result no cavity left to distinguish either it in pseudocoelom or coelom. So this will be acoelomate. This way, all the cavity is filled by parenchymal tissue. The word comes from the parenchyma in plants, which is also a space filling tissue. So this is the parenchyma or parenchymal tissue. The function of this parenchymal tissue is definitely in the absorption of the nutrient and distribution of the same, right? So in acoelomate, this is the example of planaria. You can see example of platyhelminthes. You can see the space or the cavity is filled or occupied by the parenchymal tissue, and the alimentary canal is filled, is present in between, and outer layer is the ectoderm or the ectoderm derived epidermis. So acoelomates are not having any body cavity, right? Coming next to the pseudocoelomate, in this particular category, cavity will be present as scattered pouches. First way to describe it. Second way, if the cavity is present, it will not be lined by mesodermal on the both side. So here we can see body cavity is present, but it is not lined by the mesoderm. But here I can show you that if you see this diagram particular, you see the epidermis, which is ectodermal in nature, right? And the muscle layer, which is mesodermal in nature. And this is the endoderm derived layer, so it is the endoderm. But in here, the cavity is present, but it is only enclosed by only one side by mesoderm derived germ layer, which is the muscle layer. But on the other side or the opposite side, it is enclosed by the endoderm. So although the cavity, the body cavity, primary body cavity is present, but it is not completely enclosed by the mesoderm germ layer. So therefore, in this case, we will call it as pseudocoelomate. Pseudo means false, right? So it gives a false appearance of coelom. Okay. So moving next to in between, to know more about the courses, you can scan the QR code and can register on the same. Thank you. So coming back to the next category, eucoelomate. U means true. Coelomate means that they are having a true coelom cavity. So the cavity will be there, and that cavity will be enclosed totally by the mesoderm derived germ layer. Now as you can see in the eucoelomate, body cavity that is lined by the mesoderm is coelom or eucoelom. That is one and the same thing. We can use interchangeably. Mesodermal layer lining ectoderm is parietal. Parietal means on the outerwards, peritoneum, and that lining internal organ is visceral peritoneum. So the lining of the organs is called as visceral peritoneum, and the lining of the body cavity or the body surface is called as parietal peritoneum, right? Example, from annelida to caudate. Not from annelida, but annelida to caudate means annelida is also having a true coelom cavity, and caudate is all the way organisms are classified into eucoelom or true coelom category. Now moving further, as I told you that the coelom can be developed in two ways. The one is the schizocoelom and the other opposite is the enterocoelom. Out of these two categories, the schizocoelom is considered as the primitive type or primitive method of the coelom development, and the eucoelom is considered as the advanced method for the coelom development, right? So as you can see, the schizocilum is formed due to splitting of the mesodermal layer. So if you see the diagram, these are the early mesodermal mass, right, present. This is the ectoderm, this is the endoderm, it is already mentioned. Fine. And these mesodermal mass then will further develop cavity inside them, like you can see here. Mesodermal pouches will form, and these pouch will further develop in this way, one from this side, other from this side, and they will completely enclose the full body cavity, and therefore this particular cavity, developing coelom, is marked here. This is the developing coelom, not the arrow which was earlier indicating. So this cavity in actual is the coelom cavity. Fine. So this is one method. So in schizocilum, mesodermal mass will be there, but in case of enterocoelom, the gut, that is the endoderm derived mesodermal pouches will form, and these gut pouches will now isolate and will again form the coelom cavity in the previous method as shown in this way. They will again form the coelom cavity, which will completely develop into complete coelom cavity. So this method will be called as enterocoelom. So schizocoelomic, either the organism is schizocoelomic, those who are having primitive coelom, and those who are advanced, they will be having enterocoelomic, right? So coming to the next parameter is the segmentation. In the previous slide, in the previous session, I have told you that segmentation, true segmentation, pseudo segmentation, metamerism, these all are some of the confusing terms. Here we will see each of the term one by one. So what is actually a segmentation? The proper definition is body is divided externally and internally into the segment. So in the first sentence, the external segmentation is corresponding to the internal segmentation. That means external segmentation is following the internal segmentation. So the body cavity will also be divided internally and will form different segments. Those segments can be termed as either somite or metamers. The later one is the preferred usual term choice, with the serial repetition of at least some organ or maybe tissue or some structure, like you can write tissue here or some structure. Now here you can see the example is the earthworm. So for example, I am drawing the schematic diagram of earthworm like this. This is the very first segment. Okay, segment will continue. I will change the color. Now this is the alimentary canal which is continuing. As Annelida is coelomic animal, so the body cavity will be here, will be the true coelom. So according to the definition, let us describe the coelom first and then the metamerism or segmentation. So body is divided externally. So if you see the earthworm, you will see annulus or the rings, circular rings on its surface. So that marks the external segmentation, and internally it divides also into different segments. So as I told that if the external segmentation is corresponding to the internal segmentation, the body cavity will also get divided into segment. So this external segmentation corresponds to internal segmentation, and the body will divide internally also. So if the body is dividing internally, that means the coelom will also divide into compartment. So in this case, now what happens? Each compartment, each particular compartment can be termed either as somite or metamer, each individual compartment. Further, what is the other requirement of the segmentation, true segmentation? That there must be a serial repetition of at least some organ. For example, if I have to repeat some organ, let us say we take example of nephridia, which is the excretory structure of the earthworm. So let us suppose that according to the definition, this is the nephridia. So nephridia will be present in each segment. Its opening will be on the outer body surface, and it will get repeated from segment to segment. So in this way, the other requirement is also fulfilling for the true segmentation, that external segmentation corresponds to internal segmentation, and the next one is the serial repetition of some structure, either tissue or organ, in each segment. So in this way, such type of segmentation will be termed as true segmentation or metamerism. So earthworm exhibit metamerism. Now let's move to the another term. So if the question comes on the metamerism, it would be in such a language that in some animal group, the body is found divided into compartments with serial repetition, that you must mark. It's an important character to identify metamerism of at least some organs. This characteristic feature is called as A. Metastasis, which is the characteristic of the cancer cell. B. Metamerism, which we have just explained. Third is the metagenesis, which we will discuss in phylum Cnidaria. It is the formation of polyp to medusa and medusa to polyform. And the other is the metamorphosis, which is associated with the larva development into the adult. So here we can see the answer goes to the second option, metamerism. Right way? Okay. So if we look further in the definition of the metamerism or segmentation, there are two further categories. The first is the heteronomous metamerism. In this, it is the primitive type of segmentation. If the segments or somite or metamers of the animal are all alike, they are all individually alike with each other, their appearance morphologically and anatomically they are similar in nature. So the segmentation is called as homomonas metamerism. Sorry, it should be like homomonas. You should correct it. Homonomous metamerism. So here we can see again from the example of Annelida only, the example is Nereis or clam worm, right? So here we can see that these different segments are all alike with each other. So such type of segmentation is called as homonomous segmentation, right? Moving to the next part. The next part is the heteronomous segmentation or heteronomous metamerism. It's very simple. We are all the example also of the heteronomous metamerism. So in arthropoda and chordates, the segments of the body are dissimilar and the body is divided into different segments, again different body regions and restricted only to certain organ. For example, head part, thorax part, abdomen part are group of segments called tegmata in insects, and also we are also having segmentation. For example, head part, trunk, we can call it as thorax, abdomen and the pelvis part. So in this way, such type of segmentation in which the segments are not looking alike, they are different. So such type of segmentation is called as heteronomous metamerism or heteronomous segmentation. Moving back to further to the definition of pseudo-metamerism or pseudo-segmentation. Now pseudo-metamerism or pseudo-segmentation you will find only in one of the phyla, which is the Platyhelminthes. Here in the example of tapeworm or Cnidaria, you can see that body is externally divided and also internally the body is also divided into different glottets or different segments. But in here, one condition is not fulfilled. That means there is no serial repetition. Further, to have a true segmentation, the different segments must coordinate with each other. For example, in this slide, in the diagram here, in which I have shown the serial repetition, different segments are not independent behaving. They are behaving in a coordinated manner with the neighboring segment. So this segment will behave in a coordinated manner with the adjacent segment. Similarly, this segment will behave in a coordinated manner with its adjacent segment and so on. So this is also a category which is required for the true segmentation. But if we look at the example of Cnidaria, each particular segment behaves more or less independently. So therefore, such kind of segmentation is called as pseudo-metamerism or pseudo-segmentation. So in tapeworm, new segments, proglottets are added from the interior, yani this part. neck region, and new segments are added one by one, and the segment as move further they get mature, and so in this way the whole body is divided into different kinds of segment. And unlike earthworm, there is no serial repetition, and unlike earthworm, there is no coordination between individual segments. So this is the example of pseudometamerism. Only example you have to learn is the platyhelminthes or tapeworm. Now the last characteristic is the notochord. As I have already explained that notochord is a mesodermal derived layer. Its purpose or function is for the endoskeletal support, and those organisms in which this notochord appears at any stage of life, such organisms are all categorized into the chordate. Now, so it's a mesodermal derived rod-like structure formed on the dorsal side during embryonic development, and during its development you can see that it develops below the hollow nerve cord and above the alimentary canal. So its position is usually at the dorsal side but below the dorsal hollow nerve cord. Now based on notochord, animals can be either categorized into non-chordates, those who in their life notochord does not appear at any stage, and those who are categorized in chordates, they are having notochord at any stage of life. Some of the chordates are having notochord throughout their life. In some category, notochord appears in the embryonic development, but later on it gets replaced by vertebral column. Those will also be categorized into the chordate category only. Okay. So moving further, notochord may or may not be replaced by vertebral column. The best example is us, in which the notochord appeared in our embryonic development, but later on the vertebral column replaces the notochord. Okay. So based upon whether it is replaced or not, in which it is replaced by vertebral column, doesn't matter whether the vertebral column is cartilaginous or it is bony, but if it is replaced by vertebral column, then it will be called as vertebrates, and in those the notochord is still persists, those will be categorized as invertebrates or non-vertebrates. Here we can see nuclei pulposi is the remnant of notochord left after being replaced by the vertebral column in us. Like if you see the structure of the vertebral column, each vertebrae stands upon each other. So the body of the vertebrae in between the intervertebral disc is present, and the central part of the disc is called as nucleus pulposus or nuclei pulposi, which is nothing but the remnant part of the notochord. Okay. So with this also we introduce another one of the basic concept called as cephalization. It is nothing but during the course of embryonic development, it is the differentiation of head and it is observed only in bilateria. That means those organisms which are having bilateral symmetry. So concentration of nervous tissue and sense organs on the anterior end of the body leads to the formation of the head. So if you see in our case only that the head, in the process of development of cephalization, in the head formation, the brain is developed in the interior part and the sense organs are developed on the exterior part. So, but all are concentrated towards the anterior part of the body. So, this leads ultimately to the formation of head, and all of the nervous system along with the sense organs are more concentrated towards the anterior part, or in case you can also call it as more concentrated towards the mouth part. So, in this way, it actually helps the organism to find its food with the help of sense organ, and so it's a advantageous phenomena. Most efficient to sense and respond to the environmental stimuli. So here we can see in the diagram that this is the example of octopus. So here we can see that this is the brain part, in which along with the sense organ, these two are eyes. The brain part is developed into the anterior region of the body, which is nothing but the cephalization and forming all together the head part. Okay. So this is a little quiz for you. On the basis of the today's session, you can answer this, and in the next slide we will represent the answers. So you have few minutes, 2 at least, to go through these questions, and on the next slide I will display the answer. Animals with outer ectoderm and inner endoderm are called. So any undifferentiated layer or an undifferentiated layer dash is present between ectoderm and endoderm in diploblastic animal. So what is that layer? We all know, we have studied. Phyla this dash and dash are diploblastic in nature. Now there are two phylums. One was displayed and the other I mentioned in the lecture. So there are two phylum only. The first, you know, Cnidaria, it has been mentioned. The other one I have told, that is the Ctenophora. Now dash is a third germinal layer in triploblastic animals. Is the third germinal layer. Ectoderm, mesoderm, endoderm. So you can guess which is the third germinal layer. Platyhelminthes to chordata contain three germinal layer and hence called which coelom? Acoelomate. In spite of Platyhelminthes to chordata contain three germinal, and hence are called dash coelom. So Platyhelminthes are acoelomate in spite of having three germ layers, and chordata are coelomate. They are also having three germ layers. So the body cavity which is lined by mesoderm is called as coelom, as we have discussed. First coelomate phylum is not Platyhelminthes, not Aschelminthes, but Annelida, which is the first coelomate phylum. If the mesoderm is present as scattered pouches, then it is termed as pseudocoelom. And the first one, which I have not described, animals with outer ectoderm and inner endoderm are called diploblastic in nature. Usually, we can see the other question, you can see phylum dash contains pseudocoelom. Some of the examples you cannot answer today, but in the upcoming session, you will be able to answer them. So for that particular question, you have to stay tuned for the upcoming session. So here are some of the questions on segmentation also, serial repetition, notochord also. So you can have a look, and in the next slide, we will display the answer. All right. So now, these are the answers. The first is the diploblastic, those who are having only two germ layers. The middle layer in the diploblastic is termed as mesoglea, which is the gelatinous layer. Coelenterata ctenophora, again coelenterata is already written in the slide as Cnidaria. The other one which I verbally told you was the Ctenophora. The middle layer is the mesoderm, and the organism which are having all the three germ layers are called as triploblastic. On the basis of coelom, the answer is coelomate, those who are having coelom cavity. Annelida is the very first phylum in which the coelom appears during evolutionary development. Pseudocoelom is again the term which is used for those organisms which are having pseudocoelom cavity, that means the cavity is scattered as pouches. Aschelminthes is the only phylum example of the pseudocoelom, and those who are having no cavity, they are termed as acoelomate, that is the Platyhelminthes, and acoelom is the answer of the next question. Segmentation, metamerism, notochord development, here are the answers you can check out, and if you have any doubt, you can reach out to us at any moment. You can also mention your doubt in the comment section. So what is the evolutionary milestone during all these developmental characteristics? That first, tissue grade of organization, bilateral symmetry, and the coelom are one of the most crucial milestone which occurs during the evolutionary development. So these are actually the basics parameter of the animal classification. In the next phylum, we will start from the phylum Porifera and so on. So to stay tuned, to get to know more about the courses, you can scan the QR code and register for the same. Thank you so much. Thank you for the time. एक जगह गलती हुई है, ये भी चला गया कि एक जगह गलती हुई।